Hello everybody and welcome to our section, BioNews, brought to you by the Vertex News. In today's episode, we are going to talk about gene therapy. But before that, remember that the health of our crewmates is very important to us. So several of our journalists are at home. Let's begin. After 30 years of promises tempered by setbacks, gene therapies are rapidly becoming a critical component of the therapeutical armamentarium for a variety of inherited and acquired human disease, like immune disorders, hemophilia, eye and neurodegenerative disorders, and lymphoid cancer. Treatments for this disease made with gene therapy recently progressed to approach as a drug status in the United States and in Europe, or are anticipated to receive approval in the very near future. In this review, we are going to discuss milestones of development of gene therapies, focusing on direct in vivo administrations of viral vectors and adaptive transfer of genetically engineered T cells. We also discuss emerging genome editing technologies that should further advance in the scope and efficacy of gene therapies approaches. 45 years ago, Theodore Friedman provided a prophetic account of the potential and challenges of using gene therapy to treat inherited non-genetic disorders. But translation of gene therapy concepts to patient care began in the early 90s, but plagued by repeating cycles of optimism followed by disappointing clinical trial results. So, in 1996, a National Institute of Health advisor panel concluded that these disappointing clinical results were due to insufficient knowledge of the biology of the viral vectors. So the panel recommended that investigators return to the laboratory and focus to the basic science underlying gene therapy approaches. Development of new vectors and better understanding of target cell sparked the second generation of clinical trials in the late 90s and the early 2000s. Over the past 10 years, further maturation of science of gene therapy, safely modification, and improvements in gene transfer efficiency and delivery have finally resulted in a substantial clinical progress. We are in the way to make gene therapy a technology very close to us in the near future. But this is not the end. Our partner Eric will talk more about this interesting topic. So it's your turn, Eric. We are with you.
We tell about genome editing. Genome editing is a direct manipulation of sequences in the genome where DNA of the cell is modified either by eliminating, inserting, or replacing some sequence of interesting in the genome of an organism. Genome editing can be performed on cells vivo on editing machinery, can be delivered in vivo to effect in situ genome editing. Homology-directed repair, or HDR, can be used to create a specific sequence alteration in the presence of homologous donor DNA template, which following recombination results in correction of mutation or insertion of the new sequence in the seed-specific manner. The genome editing landscape changed in 2012 with a seminal discovery by Donna and Charpentier, who showed that a bacterial defense system composed of clustered regular interspace short palindrome repeat CRISPR associated 9 nucleases can be efficiently programmed to cleave DNA at site of interest simply by designating a specific short guide RNA complementary to the target site of interest. The rapid technology advance in genome editing has made hereditary germline editing is a realistic possibility. Defined as manipulation of germ cells, gametes, zygotes, or embryos with the intent to generate a new human being with the ability to pass on the editing genes to future generations. Currently, there are many restrictions in several countries, such as lack of economic resources. Some signs that genomic editing is not approved.
Now, we brought you some experts to the subject: Diana Reyes, Gabriel Villanueva, and Axel Villegas. So please give a great welcome to our first expert, Diana Reyes. It's your turn, Diana.
Hello everyone. Today I'm going to talk about gene delivery in vivo. This technique involves the injection of a virus with a therapeutic gene directly in the patient's body. This means that first, a virus is modified in the laboratory so that it cannot reproduce. Second, the therapeutic gene is inserted in the virus, and that is called viral vector. And then the viral vector is injected in the patient's body, making all the changes that we want in the patient's DNA. Some of the cases or problems in which this technique has been proved are hemophilia A and B and an inherited blindness. In the first case, some adeno-associated viruses were used, and the results showed decreasing bleeding in patients with hemophilia A and B. And in the second case, inherited blindness, the treatment, which has been approved by FDA, so now direct injection of adeno-associated viruses is being pursued in clinical trials. So what do you think, guys? Is this treatment or technique effective? Let us know your opinions in the Facebook section. Thank you!
Hello, my name is Gabriel. I will tell you about ex vivo gene delivery via cells engineering. Okay, monogenic blood disorders and cancer immunotherapies, hematopoietic stem cells, the clinical application of gene therapies targeting HSCS derived from the success of allogeneic bone marrow transplantation for many genetic, immunodeficiency and blood cells diseases. The ex vivo approach entails the transplantation of autologous stem cells in which an underlying genetic defect is alleviated or correct. For example, adenosine desaminase deficit in several combined immunodeficiency, B hemoglobin deficiency or structural alteration in hemoglobinopathies. Autologous transplants have an advantage over allogeneic transplants in that they do not require an histocompatible donor. They avoid the immune complication of GVHD, and they eliminate the need for administration of immune suppressants. Despite relatively low HSC traduction efficiency with G-retroviral vectors, gene-modified T lineage cells were able to expand and fill the empty T cells compartment, improving immune function despite minimal levels of gene-corrected cells in other lineage. Thank you.
CAR therapy. Engineered T cells are emerging as powerful medicines for cancer. Chimeric antigen receptors are synthetic engineered receptors for antigen which in a single molecule reprogram the specific function and metabolism of T lymphocytes. They consist of an antigen binding domain either from an immunoglobulin molecule or a T cell receptor fused into intercellular signation domain, created expandable antigen-specific T cells that bypass the various and incremental kinetics of active immunization used to prime endogenous T cells. T cells that can eradicate systemic cancer in immunoglobulin cells, and CRT cells that have a large impact of treatment on certain cancers. And this success provides a function for future T cells, based for therapies from other cancers and other diseases, such as autoimmune disorders and AIDS. Now we have the conclusions. Gene therapies will be the most complex drugs ever developed, building on the demonstration of therapeutic efficacy in proof of concept clinical studies conducted in academic settings. Gene therapies are now undergoing accelerated clinical and commercial development. They are in transition from academic-based cottage industry to an industrial drug development pathway, relating on the partnerships with biotechnology and pharmaceutical companies, whose expertise in manufacturing and scale-up. This will be required for these therapies to have a broader impact. The potential of gene therapy to provide durable benefits to human health, explained by scientific advances and clinical success, over the past several years. This justifies continued optimism and increased efforts toward making this therapy part of our standard armamentarium for treatment of serious human disease. That's all. I hope that you enjoyed the presentation.